Hello, this is the first of several podcasts on how to use R and RQTL to do what's known as QTL analysis. QTL stands for quantitative trait loci, and a QTL is a region of one or more chromosomes under which or at which there are some number of genes that are responsible for a specific phenotype or physical characteristic. So we're going to look at a data set of data, and this is a CSV, comma separated values file. We're going to look at a data set from the Jackson lab that was done by breeding some mice and looking at the phenotype hypertension. So the name of the database here, the name of the data is hyper2.csv. And as you're looking at the data set there, what you see in column A is blood pressure, and that's a systolic blood pressure, so that's 109.6 millimeters of mercury for that first mouse there. You notice the first mouse is male. I think in this data set, all of the mice, yes, these are all male mice. Okay, so that's helpful to know. So we have the blood pressure, we have the sex, we have the parental grandmother, and that one there tells us a little bit about where this mouse particularly came from. I think these are all ones, so this is a pretty simple data set. So fundamentally there, the only thing that's really of interest in terms of phenotypes with this particular data set is the blood pressure. Okay, following that, what we have here is a genetic marker, and a genetic marker is a short sequence of DNA that has been identified in terms of its location on a chromosome. So there's a marker called D1, and the one there refers to chromosome 1. MIT, that means this marker was identified or um, located by Massachusetts Institute of Technology, and the marker has a number 296. The one there says it's on chromosome 1, and the 3.3 there is the location on chromosome 1 measured in centimorgans, okay, which is abbreviated C capital M. Okay. Each row here represents a mouse. So with this particular database, let's scroll down and see how many there are. It looks like there are about 250 or so mice in this study. We'll find out exactly how many in a minute. And what you should see here is for mouse number 1, okay, at that marker D1 MIT 296, the mouse is heterozygous for genotype, so has the AB genotype. And if I look at my genotypes there, I see I've got homozygous dominant, I've got heterozygous, and I'm not sure I have any BB in this. I don't think I have any homozygous recessive in this particular data set. We might indicate that anyway, just in case. Okay, I see a lot of AAs and a lot of BBs here. So when you're doing a QTL analysis, it's really useful to make sure you're looking at the actual raw data. You will need to look at the raw data to get some sense of what's happening. You should also notice that there are dashes in a lot of these things, and a dash represents missing data. So there's, for a lot of these mice here, we don't have any genotype data at a particular marker, and we'll take a look at that when we go there. Okay, I'm going to go ahead and close that data set. We're probably done with that, and I'm going to get myself in R. Now I'm going to do this interactively. But you should be able to, you should know by now that you can take all these interactive commands and put them into a script with all the appropriate documentation. I'm going to go ahead and open up what my script looks like. This was one done by Keon Lee and Randy Smith at the Jackson Lab back in 2007 with a little bit of modifications by me. And notice that this is a well-documented file. Okay, so we're going to go ahead and start, and it's going to sort of look, I'll give you sort of a scroll down so you can get a little bit of idea of what it's doing, okay, in terms of length. And I'm going to ask you to be writing a script that looks like this, okay? It looks pretty complicated right now, but we're going to walk through this in a couple different steps, and hopefully you'll be fine with that. Okay, so let's go ahead and get started, okay? One of the things that I like to do first is to sort of make sure everything is all cleaned up. And the RM command there, I'm not going to put any documentation in here, by the way. I'm just going to talk about it, okay? And you should be taking notes and writing down what all these commands do, okay? RM, parentheses, list equal ls, bracket, 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 okay? Cleans up my environment. And if I do an ls, bracket, 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 or parentheses, parentheses, and I do that, I should get nothing in there. So the data, everything's cleaned out and ready to go. Okay, because I'm doing QTL analysis, I need to load my library. And if I had not done that already, I need to install that QTL. I'm not going to do it now because mine's already been installed. You should have already done that. Okay, so if you say library QTL and nothing works, you need to go ahead and install the package. Okay, but library QTL gets things going. I like to go up, and by the way, I have on, I'm not sure where I put it, my messy desktop. Okay, there it is right there. Okay, there is a folder on my desktop called Hyper, and in that folder is the .csv file. Notice there's a lot of other stuff there because I'm going to go ahead and delete all that. Okay, this is stuff that I've already run. Okay, so basically I've just got my script, and that all you should have at the beginning is your data set. Okay, so make sure you do that. Okay, I'm going to go up for Windows people. You go up under File, and you can find your working directory. For Macs, you go under Miscellaneous and you change your working directory. And I'm going to go on my desktop. I'm going to find the Hyper um, folder, and I'm going to say open, and I'm going to want to check to make sure I'm in the right directory. So I'm going to say get working directory, parentheses, parentheses, and indeed it tells me I'm in users Gotwals desktop, and I'm in the folder Hyper. Okay, the first thing we need to do is load the data. I'm going to call my data set Hyper. We could call it anything you want. The old notation for R is these symbols here. Okay, but you can also, if you want, just use an equal sign. Okay, so the older folks use that notation. I tend to do that as well, but you can just use an equal sign. All right, what we need to do is we need to read the cross. Okay, this is mouse strain breeding data. So we're crossing two strains of mice. Okay, and I need to tell it that it is a CSV file. I need to tell it where the file is, and it's in, users, Gotwals, desktop, and everything's got to be spelled right, okay? Hyper2.csv, hyper, and hyper2.csv. So you have to tell R what the name of the file is, okay? Now, this is where you would have needed to look at your data set so you know what you're talking about. You have to tell it what the genotypes are. And what I'm saying is there's a list of genotypes, and the C command there represents what's going to follow now is a list. So as a parentheses, okay, I have in quotes AA, comma, in quotes AB, comma, and I don't think I have any BBs, but I'm going to put it in there anyway. Okay, so that says the genotypes that are in this data set are AA, AB, and BB. Okay, now you remember that there was a lot of missing data in there, so I have to say, okay, not applicable strings, meaning these are characters, are equal to double quote, hyphen, double quote, because if you remember in the data set, all of the missing data was represented with a hyphen. So na.strings equals double quotes, hyphen, double quotes. By the way, you notice in R, when I'm doing two-word things, a lot of these are separated for readability with a period in there, okay? So you need to make sure you know that, okay? And then I have to tell it what alleles it has, and that's equal to C. And what alleles do I have there? I have an A allele and I have a B allele. Okay, and if you don't have enough parentheses in there, it's going to yell at you. Okay, so I'm going to hit a return. Okay, now you get a little bit of data. Okay, it says it read the following data. You have 250 mice, there's 174 genetic markers, and there are 3 phenotypes. And if you recall, the 3 phenotypes were blood pressure, sex, and parental grandmother. It also tells you that it is a back cross, so you should know what a back cross is. Okay. Now, notice I also got a warning message. So it says some of these markers on chromosomes 1, 4, 11, 15, 16, and 17 are sort of sitting right on top of each other. So we're going to run, and it's telling me here, by the way, a warning message is something, it's just a warning. It means everything should work fine, but you should be paying attention. It's telling me to run jitter map here with no arguments in it. And jitter map just sort of jiggles the chromosomes a little bit and sort of gets the markers from sitting right on top of each other. It hurts the data a little bit, but not too much. Okay, so oops, I have to jitter map. Pardon me, I have to jitter map. How about if I jitter map the data set? Okay, okay, there we go. Okay, so I jitter mapped it, and now, okay, so it's telling me that the data set is a cross data set. It's too complex to print, so they're just going to provide a summary. And once again, you find out there are 250 individuals, 3 phenotypes. All 3 phenotypes are 100% phenotyped. Okay, we know everything about them. There's 20 chromosomes. Autosomes is 1 through 19, and there's the next chromosome. Total number of markers is 174, and they show you that how many markers are on each chromosome. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And you also notice there that the genotypes are AA and AB. There is no AB, there's no BB, there's no homozygous recessive genotype in this data set. Okay, so we have some knowledge about our cross here. It's it's a good thing to remind yourself some of the names of the columns. Okay, so I can say names hyper, and now I'm going to do a $ there because I only want to see the names of the phenotypes. Okay, so I'm going to say names hyper, and the $ there says only show me one category. Okay, and there I see blood pressure, sex, and parental grandmother. Okay, if I did names hyper, okay, I would see there that I have genotypes and I have phenotypes. If I say names hyper $geno, let's see what I get there. Okay, I get 1, 2, 3, 4 through 19. I see all the chromosomes. Okay, so that's how you do that. I can also do things like, okay, number, N is number, so the number of phenotypes is that, number of individuals, okay, is this, so if I need these numbers for something specific, I can do that, number of chromosomes. Okay, I can see all these things. There's 20 chromosomes. What else do I might want to have there? I can make a table of my hyperdata. Let's try this and see what this does. Notice that I'm parsing down a little bit. What do I want there? Okay, something like that. Okay, I can make a little data table that says that there are 250 males. Okay, so I can make these little data tables. Okay. All right, what do I want to do first? Okay, first thing I typically would like to do, I want to just take a look sort of at a plot of this data. And I'm only going to want to look at, because all I care about is the first phenotype, which is blood pressure. Okay, I'm going to say I only want to look at, let's do a plot of column 1 phenotype, which is blood pressure. Okay, so we're going to do that, hit a return there. Okay, and I get some basic information there. Okay, first thing I get up in the top left-hand corner is it shows me all 174 markers. Okay, and so that's on the x-axis. Okay, and it shows me 250 individuals on the y-axis, And then it also shows me the, and you can spread this out a little bit so it'll stretch a little bit. Okay. And what we see here is all the missing genotypes. You notice that um, a lot of the chromosomes have a lot of missing data, and that's okay. That's typical of what you get here. Okay, over on the top right, you get a genetic map, and what you're seeing here are all the markers. Each of these little tick marks represents a marker. So if you notice here on chromosome 1, there's 22 markers. If you notice on, I think, what is it, chromosome 4 here, the markers are pretty tightly packed together. They certainly are over here on chromosome 17 as well. And what that means is you've got a bunch of markers that are sort of sitting on top of each other, and that's why we use the jitter map command to sort of just move them out of each other's way, so we get a little bit better statistical analysis. Okay, on the bottom left here, you see our phenotype as a histogram. And so we're looking at blood pressure here, and we're looking at frequency on the y-axis. So you notice that we get a nice bell curve here. We have a nice normal distribution, which is really pretty important, that we have a pretty normal distribution to be able to see things here. Okay, if I wanted to, I could hit the up arrow. Let me get back into my console, hit the up arrow, and go in here and look at a plot of column 2. And you don't see very much because all of these are males. Or I could go up here and I could say, excuse me, okay, I could go up and I could say 1,3. That would show me the blood pressure and the parental grandmother. Okay, and go from there. And again, parental grandmother, there's nothing to see there because they're all ones. Okay, so there's not much of a histogram there. But just know that you can look at more than one phenotype at a time. Okay, so that's the first thing I've done there. And again, I can also, if I just want to see these things individually, I could say plot map, hyper, okay, and I get a nice genetic map. Okay. Remember also that I can say, if I want to put this to an output file, I can say this is called genetic map.jpg. Okay, and now let's turn that on, and now I say plot map hyper. Okay, and then if I turn my graphics off, because now so I get back to the screen, I turn my graphics off. If I now go look at my, okay, there I have my genetic map displayed as or saved as a JPEG that I can now open up and put into a technical memo or something like that. So remember that you can do that. One of the things you want to remember, though, however, is to turn the genetic map off. So don't forget to do that. Okay, sometimes what I want to do is I'm going to create what's called a new map here. And what I'm going to do is create an estimated map to compare with the original genetic map. So what this is doing is saying, okay, based on the data I have there, let's create a genetic map that we would expect to see, and we can compare that to the one that we actually had. So I'm going to go ahead and do that, and it's now been done. And now I'm going to say plot map, and I want to see the original map, and then I want to see the new map. So let's see what this does. Okay, and there you see that. This is a pretty complicated looking thing here. So let's pick some easy examples. This one actually isn't all that great. So if I look over here on chromosome 9, I have the actual map on the left-hand side and the estimated one on the right-hand side. And what you should notice with chromosome 19 is they're pretty much, yeah, they're lined up pretty well. Okay, so the estimated map and the actual map, for at least for chromosome 19, are statistically pretty similar. For some of these, like chromosome 1, they're really not similar at all. So that's going to be a little bit of a consideration. What you sort of hope to get here is a comparison of genetic maps where you have nice square boxes and all the lines are pretty straight across. So basically what this is doing, it's sort of like a percent error calculation. You're looking at this as a diagnostic tool. So again, what the estimated map says is, here's based on the data that I have, this is what I would sort of expect to see. And now if I actually plot the raw data, this is what I get. And again, another analogy for this might be, you got a bunch of XY data, you plot the raw data, it sort of looks linear. You now do a linear regression and you superimpose the line of best fit over the data, and that shows you what your data should be if it's statistically important. Okay. All right. One of the other things we want to do. Okay. So that's sort of a diagnostic tool. Okay. What we're going to do now is we're going to say, I'm going to generate what's called a recombination fraction plot, and I'm going to create an estimated one. Okay, so I'm going to say hyper is now this estimated chart, and now I'm going to plot that. And this one will be a little tough to take a look at. Okay, so this is a fairly complicated looking chart. What you see on the x-axis here are the number of markers, and what you see on the y-axis here are number of markers. So what I'm looking for here is, notice that down the diagonal, okay, I have sort of a red line here. Okay, red is a hotspot, and what that means is, obviously I'm comparing, let me find a place to go here. Let's go right here. So here, for example, okay, right here where my mouse is, matching chromosome 5 against chromosome 5. I would expect to see some red there because chromosome 5 is chromosome 5. On the bottom part of this chart, it's measuring what's called a LOD score, which you'll hear more about probably in the next podcast. That's the statistical measure we're looking at that says there's a significant QTL on a chromosome for a particular phenotype. And sort of what I'm looking for here are red hotspots. If I have some red hotspots there, that means some of the data might be a little misread. Maybe there was when we were phenotyping or genotyping this data, a little bit of dust got on the experiment. So I'm looking for red hotspots sort of in this LOD region down here. I've got a few down in here on chromosome 4, but nothing to worry about. I don't see any particular hotspots over here. On the top here is what's called the recombination fraction, and I've got some hotspots there, but those are okay. Really, so I'm using this as a diagnostic tool to make sure I don't have any hotspots down here on the LOD score. So far, these estimated maps that you've made so far are really looking sort of at diagnostic. So hopefully when you do your data set, you won't have any red hotspots down there. If you do, at this point, you won't know what to do with them. But what you would say is, okay, there's a chance that I've got a little error there, and I need to be a little skeptical of my results. Okay, likewise, in terms of diagnostics, I'm going to run a program called Calculate the Error of the LOD Scores, and I'm doing that on the Hyper data set, and I'm going to set an error probability of probably 0.01, and you can change these numbers. So again, what this is going to do is this is going to look at all of the genotypes and see, are there any ones that look like they might have some statistical error with a probability of 0.01 error. And oops, did I type something wrong? Oops, yep, I did. What did I do wrong there? Okay, I typed in log, I didn't type in log. Okay, it's LOD, not LOG. Okay, okay, there we go, and it's calculating that now. Okay, and it is the case that we are only, I keep typing log, we're only interested in LOD scores, okay, that have usually a cutoff, sometimes 3, sometimes 4. I'm going to put 4 here. So I'm going to say only show me those LOD scores that are 4 or higher. Okay, and so there I get a long list. Okay, so there I get a long list. So, okay, what am I seeing here? Okay, I'm seeing, okay, on chromosome 1 for this particular marker, there's an error score, and it's a lot of data there. I really don't want to have to look at that. Okay, so what I'm going to do is I'm going to say plot some of that data. So I'm going to do a plot geno, and I want to look at my data set, and I want to look at chromosome 3 in this particular case, and I want to look at individuals. Okay, um, let's see, what do I want here? I want to look at individuals, mice 130 through 150. Okay, and there I see, so for chromosome 3, okay, what I'm seeing there is where my errors are. So this is a complicated looking chart. All of these will be. So likely errors are indicated by red squares. Um, uh, for uh, for most crosses in a back cross, okay, genotypes AA and AB are represented by the white circles and black circles respectively. Okay, you probably, again, so far all I've done is some diagnostic work, and just to sort of make sure my data set is relatively clean. This is a pretty clean data set, so you shouldn't expect to have too many problems in there. Okay, at this point I'm going to end. This is going to be part 1 of this conversation, and so I'm going to stop this recording and we'll see you in part 2.